Hi everyone, welcome back to Useful Genetics. This is lecture 1E. We're going to talk about what makes some parts of our genome, some DNA sequences within our genome, be genes that function in producing everything we need to be the organisms we are, whereas the rest of our genome is just basically inert. We'll talk about RNA and protein very briefly, what these molecules are, and we'll talk about genes as informational entities in DNA and the informational processes by which the DNA sequence in a gene becomes usable information that results in a functional protein. So first, RNA. RNA is, like DNA, a nucleic acid. It's made of subunits that are very similar to the subunits in DNA. The backbone is slightly different. The backbone of DNA is deoxyribose. The backbone of RNA is a molecule called ribose. As the name indicates, the only difference is that there's an oxygen on ribose that isn't in deoxyribose. One other difference between DNA and RNA is RNA uses one different base. It still uses A and G and C, but instead of T, it uses the base uracil, a U. So at positions in DNA would be a T, the corresponding position in RNA, oh, you can't see that one, let's do this one. This would be a T in DNA, this will be a U in RNA. U, like T, pairs with A. So if this RNA were to form base pairs, the U would be paired with an A. Let's draw that right. RNA is not usually base paired. It's transiently base paired with DNA when it's being synthesized, and it may form base pairs for various functions. It can fold up on itself. Different parts of itself can base pair with each other if the bases are complementary. It can also form transient base pairs with other RNA molecules, in particular with short segments in the transfer RNA molecules that actually decode the sequence for protein synthesis. Now, protein is a very different kind of molecule. It's not a nucleic acid at all. It's still a polymer. It's an informational polymer. It consists of subunits that are similar in their ability to form a chain, but have different properties. And these subunits are called amino acids. We'll talk a lot more about proteins in Module 3. For now, all you need to know is that the proteins are the enzymes and the structures. Almost all of the working parts of the cell are protein. Now, as I said, most of our genome isn't genes. Genes are only a small subset of our DNA. And if you just looked at our DNA, any segment of your DNA, you'd have no way to tell that it did or didn't encode a gene. If you look at a piece of DNA, it just looks like DNA. Unless you examined the sequence and analyzed the sequence, you couldn't tell this was a gene. Genes are informational entities, not physically distinct from the rest of the genome. And their informational properties that make them genes are, first, that they have signals called the promoter and the terminator, which are short sequences that direct proteins to carry out the process of transcription, to make an RNA copy of this part of the DNA. And typically, they also have, within the sequence, between the promoter and terminator, they have sequences that specify specific amino acids for translation, for formation of protein, including they have signal sequences that say, make this into a protein. So this is by far the majority of genes. Some genes, however, encode functional RNAs. That is, these are RNAs that don't serve to make protein, but are functional in their own right. Most of these are enzymatic components of the protein synthesis machinery, the ribosome, or they are the adapter molecules that decode the RNA sequence and connect it to the amino acids that are going to be inserted. Now, here's a diagram to give you some sense of how these regulatory signal sequences act. Each of these signals is a short sequence of bases. And in this diagram, again, using what we said about representation, you'll see two lines representing double-stranded DNA with the crosshatch marks to help remind us that these two lines represent DNA. This oval represents RNA polymerase. That's the enzyme that's going to carry out transcription. That is, it's going to synthesize the RNA using a DNA template. And it's recognizing two signals on the DNA. So first, it recognizes a sequence that's effectively a start sequence. That's the sequence called the promoter. And the promoter tells RNA polymerase, start here to make RNA. RNA polymerase then proceeds along the DNA, and as it does, it makes an RNA copy of the DNA. I'm sorry, I can't do a better drawing of this. And it stops when it comes to a sequence called the terminator. And this is another short regulatory sequence that tells RNA polymerase this is the place to stop making RNA, to disassociate from the DNA, and to release the RNA from the DNA. So this RNA is actually not connected physically to the DNA once it's finished. So we have two signals, a start here signal and a stop here signal that are recognized by RNA polymerase. Okay. Here's a different drawing. You've seen this drawing before, representing the same process. So we have the two strands of DNA shown base paired here, and this diagram shows that in fact the two strands come apart. They're unzipped by RNA polymerase so it can make a complementary RNA to one strand. This also lets me point out that the promoter has a second function. In addition to telling RNA polymerase where to start on the DNA, it also tells it which direction to go. And it does this basically by telling RNA polymerase which strand to use. If RNA polymerase uses the bottom strand in the drawing, it has to go this way because RNA is synthesized from its 5' end to its 3' end on a DNA strand that runs 3' to 5'. So that's the bottom strand. If RNA polymerase were to use the top strand, it would have to be going in that direction. And so a function of the promoter is not just to say start here, but to also say either start here and use the bottom strand, so you're going that way, or start here and use the top strand, so you're going this way. And again, the terminator tells RNA polymerase where to stop. Now let's add on to this process the signals that control protein synthesis. So now we have two more signals, another start signal, the start codon it's called, and another stop signal, the start codon. So each protein coding gene has two start here signals. It has the promoter, which is a signal in the DNA, and it has the start codon, which is a signal that acts in the RNA, but in fact that is coded also in the DNA. But it's recognized in the RNA by the ribosome, the protein and RNA factory that will synthesize the protein. There's also two kinds of stop signals. There's the terminator that we already introduced that tells RNA polymerase where to stop. And there's a signal in the RNA, it acts in the RNA, it's recognized by the ribosome, tells the ribosome to stop here, stop making protein. Again, this sequence is coded, it's specified in the DNA, but it acts in the RNA. So we can make this happen. RNA polymerase, the ribosome binds to the start codon. It proceeds along the messenger RNA from its 5' end to its 3' end, and as it goes along, it synthesizes amino acids, assembles amino acids into the polymer of a protein, and it stops when it reaches the stop codon. The order of the bases within the DNA determines, of course, the order of the bases in the messenger RNA, and that determines the order of the amino acids in the protein. We'll talk about this in the next video. So here's a question. What does RNA polymerase do when it encounters a stop codon? Lots of students find this a tricky question because the processes are so confusing. The correct answer is, of course, that RNA polymerase doesn't care about stop codons at all. Stop codons are signals that are read in RNA by the ribosome. RNA polymerase sees stop codons as just like any other component of the DNA, and it just keeps going. Now, what we've done, we've talked about genes, that genes are basically informational entities in our DNA. Sometimes they specify functional RNAs, like parts of the ribosome. Most of them specify messenger RNAs that code for proteins. And genes are identified by the cell by the presence of regulatory signals, short sequences in the DNA that tell RNA polymerase to make an RNA copy, and short sequences that act in the RNA to tell the ribosome where to start and stop to make a protein. Coming up next, we're going to talk about how this coding for proteins actually works. I hope to see you there. Hi everyone, welcome back to Useful Genetics. This is Lecture 1F, where we're going to be talking about how the information in DNA is used to code for proteins. And coding is really very much, it's a very informational word, but it is very much the right word for what happens. So we'll talk about how messenger RNA must be decoded to a different language in protein synthesis, from the language of the bases, the nucleotide subunits of DNA, into the language of amino acids, the subunits of protein. The genetic code isn't the DNA sequence. The genetic code is the code book that explains how the translation from one language to another is going to happen, just like any other code book or a foreign language dictionary. The actual translation is done by molecules called transfer RNAs. Now, we've said several times that the order of the bases in the messenger RNA specifies which amino acids are going to be joined to make the protein. The genetic code is the specification, the connection between bases and amino acids. And it's read in groups of 3 bases. So here we have part of a messenger RNA. Here we have it split into groups of 3 bases, which are going to be, these are the codons. Remember, we used the term codon in the context of the start codon and the stop codon. The start codon is always specifies the amino acid methionine. The stop codon doesn't specify any amino acid at all, and that's why translation stops at this point. Each of these codons corresponds to a particular amino acid, AUG with methionine, ACG with threonine, etc. Now, molecular biologists communicate the code usually in various tables. Here's an example of a typical genetic code table where the rows here, this row specifies shows all the codons that start with A. This row, this column shows all the codons whose second letter is C, and then this box is all the codons that start with AC, and any of the four third position bases all specify threonine. That's not always the case, but it is in a number of cases. And you'll see the importance of this when we talk about mutations in Module 2. Now, the cell, as I said, cells don't use a genetic code table. The table is just something that geneticists have come up with. In the cell, the interpretation of the code is done by transfer RNA molecules. So again, here's our messenger RNA with the division into codons marked off for us. Here's a typical transfer RNA. This is transfer RNA glutamate, so we'll write its name as tRNA glue, and it has a glutamate amino acid attached to one end. This is an RNA molecule. Remember we talked about how RNA molecules can fold up and different parts of them can form base pairs with other parts. And that's how what starts as a stretched out linear RNA folds into this complicated structure. The key feature of this structure, apart from its particular amino acid, is the presence of a set of 3 unpaired bases called the anticodon, which are complementary to the codon for methionine. So the transfer RNA brings the glutamate to the messenger RNA inside the complicated structure that I've been referring to, the ribosome. So the ribosome is the big protein synthesis factory. And in that factory, glutamate will be added to the growing chain of amino acids specified by this chain of bases, the messenger RNA. Now, here's a second drawing with showing a simplified transfer RNA for each amino acid. There's a different transfer RNA for each amino acid with the appropriate anticodon. So this is the methionine transfer RNA with its anticodon, the threonine transfer RNA bringing threonine to base pair with the complement of those 3 bases. So this is how the genetic code is used to translate the base sequence of a messenger RNA from the base sequence of a gene into the amino acid sequence of a protein. Now, a key concept for thinking about how genes specify proteins is that of reading frames. So, in principle, in a DNA sequence, every 3 base sequence is a potential codon. I've marked off this sequence here in groups of 3, which are all potential codons if this happened to be a gene. But in fact, of course, there are 3 ways to read this sequence, depending at which base you start. So you could be reading in this reading frame, or in this reading frame, or in this reading frame, starting with different groups. So we have 3 potential reading frames. Going in this direction on this DNA strand. We have another 3 reading frames in the other direction, read from the other strand. Now, I'll show you in the next module how geneticists analyze reading frames, but to the cell doesn't get confused by this. And that's because in the cell doesn't translate DNA. The cell translates messenger RNA. And the only things that are considered by the ribosome are sequences that have a promoter so that they can be transcribed. And then they are potentially translatable. So again, here's our sequence with its reading frames. But here's a messenger RNA that's been translated from this DNA sequence. Now, this already eliminates 3 reading frames from consideration because we've only got 1 strand, which means that the ribosome will have to move in that direction. Now, to the ribosome, once it encounters a RNA that could be a messenger RNA, what it looks for is a site, a short sequence called a ribosome binding site, which is always very close to the start codon of the gene. And the ribosome binds at the ribosome binding site. There's the ribosome. And it moves along the RNA until it encounters the first AUG. And that AUG sets the reading frame. From that AUG on, the sequence is read in groups of 3. Oops, back. In these groups of 3, any other methionine codons, any other AUGs, are just treated as methionine codons. They're not treated as start points at all. Furthermore, stop codons are recognized only if they're in the same reading frame as the AUG that started synthesis. Any stop codons that are out of frame are ignored. Any out-of-frame combinations of any kind are ignored by the ribosome. It only sees the reading frame that's set by the AUG that it started with. So what we've done, we've talked very much in the language of information. We talk about coding, we talk about reading, we talk about translating codons or words. So really, literally, genes do encode proteins, going from one language, the language of nucleotides, to the language of amino acids. And the code is read in words that are groups of 3 bases. It's as if in our language all the words were 3 letters long. The genetic code is the code book that translates the language. We'll talk later about how geneticists use the code book, but to a cell, the code book is physically instantiated in the transfer RNA molecules that bring the amino acids to the codons of the messenger RNA in the ribosome. We talked about reading frames and how, although there are many potential reading frames, the ribosome knows which reading frame to consider because A, it's only looking at a messenger RNA, not both strands of a gene, and it uses a ribosome binding site and the first start codon, the first AUG in the messenger RNA, to tell it where to start. This sets the reading frame and determines how the messenger RNA will be translated. Now, coming up next, we're going to talk a bit more about the regulatory interactions and complications involved in genes becoming proteins. I hope to see you there.